And alpha hemolysis is an incomplete lysing of the red blood cells. And in that incomplete lysing, what happens is the colony forms a green area around the colony. So it's partially lysed the blood cells, but hasn't completely lysed it to the point where it's clear. So you end up having this green look to it. And it's really hard to see on here, but I'm going to pass some plates out in a second when I get done talking about all three, and you may be able to see it better on the actual plate. But it does, you'll see a colony and it will have a green halo around it if it produces alpha hemolysis. Our next type of hemolysis is the beta hemolysis. And a beta hemolysis is a complete lysing of the RBCs. And it results in a clearing zone around the actual colony. So you can see how these red cells have been completely destroyed. And in this right here, there's a clearing around the actual colony. So there's no red cells left there. And these bacteria produce beta hemolysins, either streptolysin O or streptolysin S. Streptolysin O is oxygen labile. Streptolysin S will grow in the presence of oxygen. And that can become important when we're working up cultures for strep throat. And when we get to our streps, we'll go over that a little better. And then we have gamma hemolysis. Gamma hemolysis means that there is no hemolysis at all. The organism does not contain the enzymes that help it to lyse the blood cells. And so our gamma hemolysis is pictured here, and you can see that it has not changed the agar whatsoever. And up here, the agar has not changed. There's no greening or no clearing. And so that's what we're looking at, not the colony itself. We're looking at the agar to see if any hemolysis has occurred around the colony within the agar. And examples of our alpha hemolysis organism could be Streptococcus pneumoniae. Our beta organisms can be Staph aureus or Streptococcus. And then there's a lot of the gammas can be Klebsiella pneumoniae or Proteus. There's a lot of organisms that have these different characteristics, but the ones that we most commonly see are with the alpha or the strep pneumo. And then with the beta is Staph aureus, strep pyogenes, or group A strep, or E. coli, Pseudomonas. Those are all some of the beta hemolytic organisms. So let's pass out our organisms of hemolysis here. And I'll just start up here with you guys. I'll start back here with you guys. So the plates are labeled as alpha, beta, or gamma. Oh, and I didn't go over the symbols, but the symbols that are used for alpha, beta, gamma are Greek letters. And so this is the letter for alpha. It's kind of the sideways gamma sign. Then this is the gamma sign where it's got the loop down at the bottom. And then the beta sign is like a kind of like a fancy B. And that's how they are generally recorded. Now with the use of computers, I think it's probably just spelled out. So look at your plates real carefully. What you want to do when you're observing for hemolysis is you want to use your light. So it sometimes helps to hold the plate up in the light to see if you see beta. If you see beta, you're going to see clearing around the colonies.
The other thing with beta, remember today, some it clears it wide. Some it's just a little tiny clearing around the colony. So you gotta hold it up. Even a little clearing around the colony where it eats all the blood away is beta. So let the light help you.
So an example of the one that's got the thin halo is like a group B strep. Sometimes those are hard to see. Sometimes you don't see them till the second day. You might not see it on the first day.
And we'll keep a set of these plates on either side of the room when you're looking at the real plates if you want to come compare to help. Because hemolysis, especially when you say the greening, and maybe a 48-hour on gamma that gets dark looking.
Yeah, cause gamma,
It
even
confused the
the
students.
gram-negative organisms after 48 hours,
sorry,
the
guys.
colony actually just gets darker on the media, and it does kind of get confusing, because what is a green? It's kind of a dark too, but you wanna make sure that it's the halo.
Okay.
So remember, I told you, don't get discouraged. We're building skills, and there are
So
some
did-
that it gets.
Did you guys see the alpha and the beta? Cause some people have trouble with the
Yeah.
alpha. Were you able to see the, oh, yep, that's right, I started in the back. Did you guys do okay seeing the greening and then the clearing?
Cloudy.
Oh, yeah, it's definitely white.
But we'll keep a set on each side. That way you have something to go back to help when you're, especially those of you who have Streptococcus pneumoniae as your organism. You'll wanna maybe compare to make sure if it looks like green like the plague. I don't know if we
Yeah,
have
so is
a
mine.
group B
Yeah.
strep. as one of
We
our-
don't have one yet,
Right.
we're gonna have to get one
Get
for
one.
this, when we get to steps.
So we may not have something that is a little layer of beta. We have some that are big, where it eats it out. Those are easy.
Notating that, or do you,
The
I mean,
hemolysis,
whenever you, or is that just, to help differentiate? Like, whenever you say this is what the organism is, it's, you know, out there.
yes, so when we're assessing the bacteria, when we're doing the morphological characteristics of an organism, you have a worksheet. And on your worksheet, it'll be divided into your medias, so you'll have a blood agar plate. And you record the morphological characteristics of the organisms on this worksheet. And so you will record the hemolysis. That was always the first thing we recorded in our lab. Even when we went to the computers, that was the first thing it brought up on the worksheet was hemolysis, and you had to record the hemolysis. It wouldn't let you move on to the next phase until you recorded hemolysis. And in the old days, you recorded it with the Greek symbols. Nowadays, I'm sure that everybody's using a computer worksheet, computer-generated worksheet, so it's probably written out. So, the next thing we're going to talk about is the actual colonial morphology. And so these
I
characteristics.
did give you a second sheet, and there's one in your book, your syllabus too.
The characteristics of each colony type are very helpful in guiding us in a direction for the identification of the organism. Because some, like Micrococcus luteus, it's always yellow. So when you see an organism that's growing on the blood plate and the CNA plate and not the MAC, and it's yellow, kind of one of the first things you're gonna think of is, oh, maybe it's Micrococcus luteus. Now, that doesn't mean that's what it is, but a lot of times when it's a bright lemony yellow, that's just what a microbiologist is gonna think. There's other organisms that do that, but that's one of the more common ones. So those particular characteristics aid us in the identification of the organism. Kind of gives us an idea of what step to take and what biochemicals to set up and things like that. And we generally observe those characteristics at 18 to 24 hours. So that's why we had you guys sub those organisms today. And then tomorrow, you're going to go get your organisms out of the incubator. You're going to have a worksheet sitting there, and you are going to assess the morphology of your organisms. And they will have been incubated for about 19 hours, 18 hours. And in these characteristics, we're looking at size, shape, margin, elevation, pigment, service, optical property of a colony. We're looking at those things, but we don't record every one of those, or your worksheet, you wouldn't have enough room on your worksheet. So you're going to pick the outstanding characteristics. And if there's nothing outstanding, then it's generally you do hemolysis, size, color, maybe the shape, and that's your identifying characteristics. And another purpose of the identifying characteristics is if you have a culture that has multiple organisms in it, and you're working up this culture on the first day, you need to give a good enough description of each one of the different colony types on that media, because tomorrow, it could be a different person looking at that plate. And if you don't make a good enough description, they're not gonna know what you were talking about when you were working things up, and then they're starting all over. If you're there, then they're constantly bothering you and you're having trouble getting through your work because they're saying, hey, what did you mean by this? And I mean, that does happen sometimes, but not constantly. So you wanna give good descriptions of the morphologies. So when we're looking at shape, you're going to look to see if it's round, rhizoid, irregular, filamentous, or spindle-like. The margin, which is going to be the actual edge of the colony, is that entire, which, you know, is it a complete colony? Is it undulate, lobate, curled, rhizoid, or filamentous? And swarming. We do have an organism that swarms the entire plate, and that's always a Proteus. And you guys will see that, but you'll see the center of the colony on the plate, and then there is just a film of growth over the entire plate. And that is called swarming, and that's generally a Proteus. The elevation of the organism. So the elevation would be looking at the plate from the side. And do you see, is it raised? Is it completely flat on the media? Is it convex, pulvinate, umbinate, or concave? The size. So with the size, we have 4 descriptors. It's pinpoint, small, medium, or large. And I've got some plates that I'm gonna pass out for you guys to be able to see the different sizes. Okay, so you're gonna wanna look at these plates and compare the sizes. And so when it's pinpoint, it's like just a little, almost like a little speck on the plate. Sometimes on the first day, you have a really hard time seeing the pinpoints. You may miss them because they're so tiny. Candida glabrata is one of those organisms. Sometimes you'll look at that plate, especially if it's at the 18-hour point, and you don't see anything on that plate at 18 hours. And then you go to look at it at 48 hours, and all of a sudden, there's this pinpoint organism on there. So in the sizes, we're generally, the gram positives are generally smaller than the gram negatives. Staphs are usually bigger than the streps. And so that,
Your practical, it's gonna be based on the size at 48 hours. So we'll give you a key that you'll study, and it'll all be based on what it should do by 48 hours. Sometimes at 24, depending on if it was
right.
full 18 hours or full 24 hours incubation, it may be way smaller than
Right.
it will be at 48.
And you see that a lot with the staffs, like sometimes, like Staph epi, on the first day, you might call it small, but then when you see it the next day, it's medium. And unfortunately, in the real clinical setting, you don't get to look at the plates all the time, right at the 18 to 24 hour period. It could be that something got set up at 11 o'clock at night, and it's still included in that day's work. So when you're looking at it the next day, it might have only incubated for 16 hours. And so what a lot of labs, we especially did it, because a lot of our work did come in on the evening shift. We only read 48-hour cultures in the morning, and we then read our new cultures in the afternoon. So you'll see the size changing from the 24 to the 48. Is everybody getting kind of an idea of what we're talking about with pinpoint versus large and?
Moderate and large, they seem to be very close. Is it going to be an issue if you call a moderate a large or large a moderate?
No, and these are the, like we said, the 18-hour, and so tomorrow that large might really look large.
Yeah, remember, if, at least you're gonna wanna do color, size, and hemolysis at
At the
minimum.
least, yeah.
And then these other characteristics, if you can tell them and it is significant that it helps you, identify, but at minimum, color, size, hemolysis off the blood plate. You're always
those are three
using the
different ones,
blood
'cause-
plate.
Right, you're always doing your colonial morphology evaluation from your blood plate.
Are these all
Well,
three?
unless it doesn't grow on blood, like sometimes you have to do it off the chocolate in case of
Right,
hemolysis.
like your
Come off the
fastidious we talked about,
Yeah.
that enriched media, it won't grow on the blood, so you'll just put NG, and then you gotta look at the colonial morphology off the chocolate.
But you will not assess hemolysis on a chocolate plate, never.
You see that clearing? Is this the one with all three, or what? Is this one large and medium?
No,
No,
This
they're
is
not
color.
It was just a blown up
we
version,
were just
and I
gonna-
have a description.
Yeah, just look at the size. I also handed out the color plate, cause I'm talking about that next. Yeah, so, and like Jean pointed out, you're not always gonna use these descriptors, but for example, the one that's concave, strep pneumo they call it the donut shape. So strep pneumo will be concave in the center. So if you see an alpha colony with a concave center, you wanna make sure you notate that, because that is telling the tech a lot. Because there aren't a lot of organisms that react that same way. There's a few other streps, but definitely, if you see an alpha colony with a depressed center, you're gonna start thinking strep pneumo, and that's gonna clue you into setting up a P disc and doing a biosolubility and all those other tests that go along with the strep pneumo. So whenever there's an outstanding characteristic, you wanna make sure that you note that. Another characteristic that's really not discussed on here is the smell of an organism. And Pseudomonas has a grape-like odor. So when you open up a plate and you get a whiff of that grape-like odor, definitely you wanna notate that. Because again, that's a characteristic that gives you a whole big picture. So then we go to the texture of an organism. So you get the texture when you go to pick up an organism and you put the loop in there. And you can have a texture that's rubbery. And if it's rubbery, it just kind of like, you go to pick it up and it leaves like the shape of the loop and everything right there in that colony. Smooth, well, a smooth colony is just like it says, there's actually not a whole lot of texture to it. If it's rough, so you'll see colonies that they can look like kind of bumpy, and when you go to pick them up, they kind of crumble. They can be dry, which again, the dry ones will crumble. Mucoid, Klebsiella pneumoniae, that's one of the big characteristics for that. When you see a lactose fermenter on a McConkey plate, and it's large and it's very mucoid, or a lot of people say it looks like snot, because it does just kind of like real stringy and runny. So that's a definitive characteristic for Club pneumo. Waxy, you'll see that with some of your Bacillus species, that they just kind of like fall apart like wax. Like if you have a candle that melted and then you go to knock the wax off, and how it just kind of breaks up, that's kind of how that does. And sticky. A lot of the Micrococcus are real sticky. And when you go to pick up a colony, to streak it to a plate or to make a gram stain, that entire colony will just pick up and stick to your loop. Surface appearance, so you might get colonies that have a wet sheen or a shiny sheen to it, which a lot of our gram negatives do. They almost look like a puddle of water. Are a very dull looking colony where there's no sheen to it at all. They can be wrinkled. Metallic, that's another characteristic of Pseudomonas aeruginosa. So when you have a plate that has a gram negative on it and you hold it in the light, it has a sheen to it and it looks like a piece of metal. And that's very characteristic of Pseudomonas aeruginosa. There's not any other organism that has that metal sheen. The pigmentation, And so that's why you guys have that second plate that says white, clear, yellow. So we look for that. We look for the color of the organism. Like I said earlier, if you see a lemon yellow, it usually means Micrococcus luteus. There's tons of organisms that are white, tons of organisms that are gray. Clear. Some of our streps, our beta hemolytic streps, are just clear colonies. So those are all factors that kind of help us and guide us as well. When you see a beta colony that's clear, you're going to think strep A, C, or G. When you see a beta colony that might be white on the first day, but then kind of has a buttery appearance or a light yellow on the second, Staph aureus. So the color is another factor. So you guys have a plate that's got like white, clear, yellow. If you want to look at those. And I think it was a pretty I put them out there, and I think they all looked at them with the size plates. But, I mean, you can see where it's distinctively different. And that really helps when you've got multiple colonies on a plate. Sometimes it's hard because you might have multiple white colonies, and on the first day, you can't tell the difference. They all look like the same colony. Then on the second day, you might see a little bitty variation in color. That's where it starts to get hard, when you have to be looking for a little tiny variation. You might see a white colony on day one, on day two, then you have to, they turn into two, and you've got to say gray white, or tan white, white white, bright white. So it gets a little funny, but you're just trying to look for distinguishing factors so that you and the next person know what you're talking about. And then there's the optical property. So is it translucent? Is it opaque that you cannot see through it at all? Or is it transparent where you can completely see through it? That you can read the type of media through it. So that's how we're going to go through our next step. We're going to pass out worksheets. And on these worksheets,
So you want your first worksheet with the number 1 through 7 that I gave you in lecture.
so you're going to have the organism. And you're going to write, we're going to start with this, we're going to do Gramstein first and let it dry.
Yes, that way,
Okay.
let's do, and I
So,
just gotta pass their trays
Let's,
to them.
before we start looking at our plates, let's go ahead and do the gram stain, because with the gram stain, we emulsify the organism in saline or water, and it takes a while for that to dry. So we'll do that first, so it can dry while we're evaluating the plates.
So you're responsible to clean your trays at the end, and then you can set your reagents right in the tray, and then just keep that at the top of your desk.
Thank you.
Oh, and then let me, that one had tweezers, let me give a little set of tweezers per table. That you can keep with it. Oh, and then I gotta pass out a box of slides. Here, you wanna pass? Oh, you got one? You wanna just pass that up? Pass this one up, I like the thicker. And then, let me give you, the slide has, I think these have the circle in the middle. Yeah, so you're gonna pass out, we'll do a box of slides per desk.
So what we're gonna do is, each of you have an organism at your table. You're gonna get a slide, and I want you to label your slide, once again, name, date, and put your initials on it so that if we don't look at them until tomorrow, we know whose is whose.
Here, you guys.
And then what we're going to do is we're gonna add a drop of saline to the slide. And then you're going to pick up a colony of the organism, and you're gonna emulsify that in the saline.
You wanna, there's salines in the middle, I just have DI water at their desk.
That's fine too, DI water will work.
You should
Doesn't
have
matter.
a tube of water that I put at each desk with a black cap. You wanna,
Oh, we gotta hand the organisms out.
here you go, here's a box of slides for up here too.
Do you want to keep the organisms in numerical order?
Yeah. Yeah, let's do.
Do that.
Yeah, we can end with 7. Well, whoever, we'll end with 7 back here because one can do 6 and one can do 7.
So we have staff on here twice. Oh no, this is number 8. First bunch.
This one, right?
So we need number 1 would be Cleb
Thank
pneumoniae.
you.
On that side, Cleb, Pseudomonas, and E. coli on those 3. And then we'll do 4, we'll have
Okay,
strep pneumo.
whoops, wait a
we'll
minute.
also do 5,
What was the first
Epi.
one? Pleb. Pleb then pseudo?
Yes. And then over here, I need strep pneumo, this table, then staph epi, this table. And then we'll have you do 7 and Chris do 6. So 6 is Staph aureus and she's Candida. Oh, what's on?
Oh, son
Do we,
I'm sorry.
oh, we don't have a Candida?
Yes, I messed up.
Well, it's okay if we do 13 organisms and then we can have a,
No, because I have a candida that we can do, we'll just not have that.
we can just say,
sub the,
let me go do the plates and get it in the incubator. For at
we'll
least
just-
tomorrow.
We'll use this one, but then we'll sub it to the blood and CNA, so that way they can see what happens on that.
Okay, and the McConkeys, just so they see it doesn't grow.
Right.
Okay. So we can give that to Leah, because she can help with Chris, see Staph aureus as well, all 3 plates as well.
We need 3.
So what you're gonna do is you're gonna put your drop of water, emulsify 1 colony in that drop of water, then we're gonna let that air dry. Once it's air dried, we're gonna fix it with methanol.
So we don't have the crayon markers.
Which one's?
And it'll-
I should have made a note to do that. Well,
Let's see. Oh, I know, you can write
Where's the
it
drop of
in
water?
pencil. These have the side, you can write it, it won't go
yeah,
with the methanol.
I thought you meant for the water.
Is it okay?
No, it has
Oh,
a
they've already
circle.
got circles,
But-
yeah. Don't write with marker.
A tiny drop.
When you write with marker, what's gonna happen when you add the alcohol to the slide? It'll wash it
Oh,
away.
yeah.
So you always wanna use a pencil. Don't use ink or markers.
Because these have the white where they could write with pencil.
Yeah.
But remember, a tip, don't use a ton of water, it'll take forever to dry. A tiny drop of water.
Are we using DIY?
You use the stuff with the black cap?
Yeah,
You don't have
yeah,
a pencil?
I gave one to every table. And just use, can they just use a loop to add the water on?
These are pretty small. They can try it.
Or do you want me to use the plastic droppers?
What I
What's it called
always
at work?
do is I use a plastic dropper, but I don't let the drop free fall. I touch the drop of water to the slide, so that way you just get a little water. Don't use a full drop.
Here you guys.
Do we have the pneumatic routines?
Can we just use the loops?
So
You could
just,
try it with the loops, but they're pretty tiny. You can
Just-
see if it'll
you
work.
know, you can just take a handful.
Did you
But
did you
just
find a
touch
pencil?
it, don't let the drop free fall, it's gonna take forever to dry then. I'll put a box back here and up there.
Yep.
Yep. Just, you wanna,
Yeah, you
you
can
only
put
need
organism,
off
date,
one
and
side
then-
for the blood, you guys, that's all. You gram
No,
stain
you're gonna
from the
put your drop
blood.
of water first.
Here you go.
Yeah, add your drop of water to your slide first. Then you emulsify the colony into the water. So you want to break the colony up pretty well in the water. You want it to emulsify.
Does it matter which one?
From which plate?
Yes.
Yep.
Which
We're
plate are
always
we
going to
using?
use the blood because it doesn't
Yes.
have any inhibitors. If you use the CNA plate, it's got antibiotics in there that are inhibitors, and it can change the cell wall
Mhm.
of the organism.
Okay.
And we don't want to change the cell wall because the cell wall is what gives that organism the ability to retain the crystal violet.
Gotcha.
you want me to
So
go get a
we don't wanna
controls tube?
compromise the cell wall, so you always use a blood plate.
And here, I'll give you each person their own QC slide that has gram-positive cocci and gram-negative bacilli. So each of you have to do your own 2 slides, one being the QC and one being your organism.
So the QC slide, you're not gonna do anything to it right now.
Grab some, now I'm just gonna,
And these are already methanol.
Okay.
I did that.
I just grabbed it from somewhere.
You said what?
That was
Do you have to like re-grab after each time you put in?
Pardon?
Do you have to re-grab or each time you put in the water
We're
or
doing 3
not?
slides?
This one?
Nope,
Or
one from
You
your
should be
blood.
able to only pick up one
Everybody should
colony
just
and
have 1
emulsify it.
gram
You're just
stain
gonna do one,
from
okay,
your
you guys
blood.
are only doing one slide from the blood plate, that's it.
Okay.
We're not doing the MAC and the CNA, only the blood plate. So you should only have one slide labeled.
Okay.
You put a drop of water on that slide, you pick up one colony, unless you've got a really tiny one, like the Candida glabrata, you're probably gonna have to pick up a few colonies,
So you
because they're very tiny.
Do I throw it out? Do I do a new one?
Did you touch the slide?
Yeah.
Yeah, I would throw it out.
Yes.
Right,
This
so the
one
QC
already
slide,
has
you're not gonna
methanol.
do anything to it right
Set
now.
aside yet.
The QC slide, we're
Crystal
just
violet.
going to stain it with our organism. And the reason why we do that is we wanna see if we have stained the organism properly. Did we over decolorize, under decolorize? You can't make an assessment of the microscopic morphology if you haven't stained it properly. So that's why we're running a QC slide along with our organism gram stain.
I use the whole circle.
Okay, so has
Did you drop the
everybody
water, right?
got their slide drying?
Here, did I? Mine's about,
Yeah.
I made it look like the QC. I tried
We
to.
need to tomorrow, since we only have.
So we'll go over the procedure tomorrow. I'll put this back.
Sure, that looks like it'll do. I guess it'll do.
You know, I don't think it's bad if we get them gram stained, we just won't get to, because we already talked about it in lecture, so I mean, it's your
Yeah,
choice.
we'll see
Okay.
how far we get along, because they really need to start evaluating all
Looking
that.
at it.
Yeah.
Okay.
Okay, so once you have your slide finished, just sit it aside, and we're gonna let it air dry. And we might stain it today, we might stain it tomorrow, we'll see. Mine's dry.
It'll probably still show, just a new.
All right, so what you're gonna do next is you have your worksheet, and you have your organism. So what I want you to do is you're gonna look at your blood plate, and you're gonna evaluate Haemophilus, size, color, shape, any other characteristic that's outstanding.
So make sure you put BA, on the BA put, G will be growth, NG, no growth. I gave you that little sheet of abbreviations. So that's the first thing you're assessing, did it grow or not grow? Then look at your colony on the blood plate. So
Okay.
can they at least do G all the way across and do
Yeah.
G or NG?
Yes, that's,
Blood oxygen.
G is the acronym for growth, NG is the acronym for no growth.
So in your case, we could have her put G on the CNA,
In
the
the
yeast, and she, since she
We'll
doesn't.
wait till tomorrow. Just,
So
just
just-
put a line right now, because you
Yeah.
don't have it. Yeah, just put a line like, or NA, put NA
Yeah,
for
put NA
MAC
for-
and CNA,
For them too.
because you don't have it at 24 hours.
So what about growth for, like in the first section, but there's no growth in the other two?
Any growth.
Any growth on the plate whatsoever?
Right.
Yeah, okay.
On
If
any
it doesn't
bird.
grow, there shouldn't be any colonies, anywhere.
Okay, what is that?
Oh, do we talk about volume yet?
No, and I was gonna say we'll do that.
Tomorrow.
Well, we could do it tomorrow or in the next step, like when we start working on I kind of thought just leave it right now as to
Growth or no
Growth.
growth.
morphologies and
No,
stuff
no,
like
growth
that.
is eight.
I agree.
And then we'll go into quantitation the next time
Oh,
on.
I think that's
Is
good.
that all right? Okay.
Yeah.
Under the Gram-Stain
Yeah,
column, is
so
that
just
the
what you wanna
GM,
do,
the
evaluate your
GC,
blood
GC?
plate.
Here, let do. I
Once
have a-
everybody's evaluated their blood plate, move on to your McConkey plate, and I'm just gonna talk about it right now, so that way, you're all gonna get done at different times. So what you're gonna do with your McConkey plate is you'll put G or NG, then you need to assess if it did grow, is it a lactose fermenter, did it turn pink? Or is it a non-lactose fermenter, and it's clear or pale?
Did I give you-
And then on your CNA plate, all you need to do is record if it grew or did not grow. And if anybody has any questions along the way, feel free to ask.
No, did I not have those organisms on the?
Yeah, growth in all three.
You think?
Have an extra one. You can put it here and show us.
Oh, okay.
Because I have,
Well, you want me to just
I mean, it's definitely not
Yeah.
pink or
because I should have an extra one, because I had extra of the
What is this?
actually 14.
If anything, I see a little bit of pink in there,
Yeah, then you can show them your abbreviate, how you would do it.
Just the VA.
I'd say, I don't know.
All right, guys, real quick.
Okay, I'm not sure.
So say I have organism number 1, and I look at my blood plate,
Yeah.
I'm gonna record the hemolysis, and since we are doing written recording and not computer, I'm gonna use the symbols, the Greek symbols. So remember, beta was the B-like, alpha was the sideways loop, gamma was the loop on the bottom. So say I have the Kleb, and I think it looks gamma. And I think it looks gray and large. And say today it's mucoid, so I will note that. Then I'll move on to my McConkey plate. Oh, and I didn't record, I needed to record
Oh.
those. On my McConkey, I'll record it as growth and a lactose fermenter. And that's how I would do it. That, so that's how you're gonna wanna record
This has
your
the CNA.
information.
so
So you can-
Okay,
You don't have morphology.
That one in there doesn't have the CNA.
all right,
Do you want
so
us to add the
the
morphology?
hemolysis, you're gonna record the
Morphology
morphology.
is under the BAP
Okay.
only.
So see how, that's what I'm describing right now, is the morphology on the blood plate of, say, organism number 1. It's gamma, it's gray, it's large, and mucoid.
So mucoid
And it
made a
grows.
distinction, that's why she put mucoid. But at least put color and size if you can't do any other descriptor that matters.
Then the McConkey I'll record as growth with a lactose fermenter, and the CNA is no growth. So that's how you wanna record your morphologies.
So and I left the hemolysis plates back here, I left the size plates, and I left the color plates. So at minimum on the blood plate, at least you color and size.
And look at it from the side, that's another easy one. If it's mounded or raised, if it's completely flat on the plate,
And I do
Those
have
are things that-
magnifiers in each drawer. The black magnifier is for your help too. Yes.
Oh, boy.
And these are tiny pinpoints. She said the glabrata. So you really have a hard time distinguishing if it's clear or white. So this first day, you're gonna probably, if you don't put it, that's okay. It might be bigger and able to see the color tomorrow. Like, anybody have it? When we, did we pass them around first day?
what?
As we passed the different tables,
Yeah,
right?
they passed around to different tables, so that way they can move down the
Right, then
sheet.
you'll see when you get to a strap, some of those are clear. They almost look like water. They don't have a color,
So
like a
who's
white or
done
off-white.
evaluating their first 3 plates? Anybody else? No?
Got
You're done?
one.
Yeah, no one's gonna get to do much on the,
And
on 7, but. So you wanna give a setback here? Oh, you're
I'll
giving.
have to wait for another set, or if you wanna, they already started.
Yeah, just raise your hand when you get done looking at the three, and then we'll come get it for you and pass
Yes,
it to
now
someone who
we're gonna
needs
pass
that.
all 7 organisms around, so then you will do the same thing for each organism.
You need another,
You don't have to gram stain, but the
No.
one you started
We're only
with.
we're only gonna gram stain the first organism that you had, and then tomorrow, everybody can look at all of them, but we're only gonna have one set of slides for each organism.
Yeah, we'll put them at the desk. You need a set? You can at least do, you gotta do all 7. So I need your plates.
Okay,
Are these it?
Is that on the
We're
conkies
comparing,
or is it? I thought that was just on the blood.
compare the.
Oh, okay. So.
For the
Here,
color,
here,
do you
do
record that on the
Canada. You
conkies?
just
No,
put the one just
On the
like-
McConkey,
You
you
just
only,
put
you're
a
gonna
line
record it
NA
as an
on the
LF or an
Mac and
NLF.
Right, so
Which I didn't.
CNA.
Okay.
the color is only on the
We gotta
blood.
send them up.
Right,
Okay.
right, so the McConkey,
Did
you're
you
gonna
guys look
record
at
if
these
it
three
grew or
yet?
didn't
Are you still
grow.
looking at them?
And then if it did grow,
Okay, here's a group.
is it a lactose fermenter? Did it
That's
turn
okay.
pink? If it did turn pink, you record that as LF, and if it's clear, it's NLF. Okay, you guys, I did not tell you the acronyms for the McConkey plate. If it's a lactose fermenter, which if it turned pink, you will record it as LF. If it did not change color, if it's clear or really pale, then it's NLF for non-lactose fermenter.
Didn't change.
So you don't have to write
So
out
that is a-
lactose
An
fermenter. or
NLF, there's no
okay.
pink at
I
all.
was like, it's not clear, but.
But I told
Well,
them beige
there's
or
no
buff.
change, it did not absorb that dye, it didn't turn
It would have to be
pink,
pink.
like,
Did you
like
guys do your
this.
3
So
plates? And I'll take your 3.
here's the example of the lactose fermenter
Oh, there they
versus
are.
non-lactose fermenter. So this, the lactose fermenter took up the dye very well, it's pretty pink.
Okay.
The other one is a pale or, it's not clear, it's kind of a palish pink because the media is a pale pink.
Yeah.
And remember, you guys, on color, what might be off-white to you might be gray to someone else, unless
Yeah.
color is distinct, like micrococcus with the lemony yellow. Color doesn't always give you a big clue.
No, it doesn't always give you a clue, but you always need to use it as a
Record it.
descriptor because it helps the next tech to look, that's looking at it.
But remember, don't
Yeah.
sweat it if you call it
Yeah.
off-white, someone called it gray.
And like I said, that, sometimes in the lab, we'd call something white on the first day and then the second day it was buttery. Staph aureus is really good for that. It'll look white on day 1 and on day 2 it'll have a real pale yellow butter color.
Lizard growth on the blood.
Yeah, glabrata is kind of small, hard to do much, so at least the minimum, you can at least say pinpoint. So, Chris, on that one. Because we don't have the other 2 plates.
So, yeah, everybody,
So,
on the glabrata, which is number, is that 7?
yes.
Number 7, go to your Mac and your CNA plate, and for your 24 hours, record it as NA, because I didn't sub the Mac and the CNA, so you don't have it to look at today. So just put NA on glabrata for the CNA and the Mac. You will not be looking at those plates today, you'll look at them tomorrow.
So I'm gonna set the glabrata next to you guys.
And I put,
That would be under.
And always in the blood plate.
Miles.
Yeah, we pick up the kind to see if it's smooth. Oh, that's right, this one's
Yep.
magnum.
You're gonna have minimal written on the MAC and the CNA. Your morphology descriptors are on the blood plate always.
It's smooth, it's the right, get it. Yeah, then we'll take
Okay. Oh, someone has it. Do you have one over there to use?
I don't know, but if I don't, I'll just grab theirs.
Okay. Yeah, just gather some of those in now. So remember, the BAP is your big time plate. See if that's one you guys didn't have. Someone else ready for a set? You had that one
Number-
already?
Which one is it?
This one is number staff epi.
I'll do it.
I'm gonna pass that over to them. Let me see this, yours. What was your guys? Club pneumo, did you guys already do club pneumonia?
No.
Here you go. Yeah, when someone,
That is,
Is he on the side?
What's
I'm saying
probably
gamma phone.
behind us.
Oh, I
So,
forgot to put that.
so you're still waiting for one?
I guess not seeing any clearing around it or
Take
any
theirs.
green. Stings.
Oh, her actual one, not the one I passed to Nicole, okay.
Beta.
Yeah, I'm gonna try that
Beta's
So
the
remember, your blood plate, you really want to have good isolated colonies on that one. That streaking is so key. The others have inhibitors, so they're probably only going to be individual colonies anyway by the third quadrant because of those inhibitors. But the blood, especially if you have a lot of different organisms. I'm gonna make a sheet for this too.
MG to HG.
So, as you get done, you guys, are you done waiting for one?
Yeah, sorry.
Yeah, I think tomorrow we'll pass them and just pass them back. So we get them in a more organized fashion done. We'll just pass them around in a structure. Because now we're doing hit or miss.
LF.
Yeah, I think tomorrow we'll do 8 to
Uh,
9 to 10 because if some get a lot done and some are
Right.
right.
So if you just put C in there,
Okay,
you just gotta
who's
put
got the Candida
no
growth?
growth over.
Yes. Wait.
Oh, okay.
Yes. We can't be on that.
If-
This one doesn't change.
You done with your toy?
Hey Jean, on the CNA, she just said put growth but no growth, right?
Yep.
Nothing else.
Right. EAP is what you do the descriptors. Now, you have a little more on McConkey because why? It's differential. You gotta say LF or NLF besides the G, if it does grow.
So no growth and on that.
Anybody want growth? But pneumonia?
Try again.
Anybody need strep pneumonia? Oh, you still do?
I have 1, 2, and 4. That's alpha.
Do you need E. coli?
Yeah. Tomorrow we'll pass them in an order
3.
around.
Oh, yeah, that is.
Anybody need
That's
E.
gonna be
coli?
more efficient. What do you need, E. coli?
Yeah, I need number 7.
So, we won't move on to anything till everybody has looked at them.
Do you wanna start just passing them in now?
Oh, we'll let them finish up this way, and then,
Don't.
and then
Yeah.
maybe at least if we can gram stain, that'll be good. Get the gram stain done, and I'll save it for tomorrow, I got the yellow thing out.
I kind of held it over.
And then I'm gonna keep these sheets here with color, LF, hemolysis, cause I know Barb will be using this table today. So if anybody wanted to take pictures or anything, plus don't forget to look at that biochemical PowerPoint on Blackboard, it shows a lot of what we're doing.
Would you, hey Jean, I got a question. Would you see clearing on here? 'Cause I don't.
Clearing, did you put it up to the light
Yeah,
and look around the colonies? What's your organism?
staff aureus. I don't
It
see
should
any.
be.
I do, I can see it on here.
I saw around the big section. I saw.
Where
you can
it's
see-
clear, it's
Well,
not.
and one other